Welcome back to the deep dive. I hope you have your hiking boots laced up tight today because we are heading straight into the woods.
We are indeed.
And we're not just going for a stroll. We are going on a treasure hunt. We are looking for royalty.
A very specific, very ancient kind of royalty, not the kind with crowns and castles.
No, this is something much more organic. We are talking about the emperor of the woods, the Amanita Jacksonii.
A truly stunning mushroom.
It is. It's this brilliant red-orange mushroom that honestly looks like it was drawn by a Disney animator. It's almost too perfect to be real.
It really is a spectacle. But what makes this deep dive so fascinating to me isn't just that the mushroom is beautiful.
No.
It's that it represents this massive biological paradox.
Right, and that's the hook. We are talking about the genus Amanita.
Which, if you know anything about mycology or even just a little bit about mushrooms, that name usually makes the hair on the back of your neck stand up.
Exactly. This is the family that contains the heavy hitters, the death cap, the destroying angel.
The ones with the scary names for a very good reason.
The mushrooms that you don't even want to look at sideways.
It's true. The Amanita genus is responsible for roughly 95% of all fatal mushroom poisonings worldwide.
95%? That's a staggering number.
It is. These are efficient, ruthless killers.
Yeah.
And yet, sitting right in the middle of this mob family is Amanita Jacksonii, a mushroom that is not only safe,
But is considered a top-tier gourmet delicacy.
one of the best.
It's like finding out that the cousin of the king cobra is a golden retriever who wants belly rubs. It doesn't seem to make sense.
It completely defies expectation.
Yeah.
And that's our mission today. We have a stack of sources here. Genomic studies from the International Mycological Association, field guides, some really interesting historical accounts of Roman emperors.
And we're going to figure out how this paradox exists.
We're going to try.
We're going to unpack the biology of this mushroom, the slender Caesar, figure out how to identify it without dying, which is a very real stake today, and look at the invisible machinery, the genomics, that makes it tick.
And we're going to look at the neighborhood it lives in. Because the genomic study we're referencing didn't just sequence the emperor.
Oh, this is the really cool part.
It sequenced the villains living next door, the pathogens and the beetle riders. It's a rough neighborhood out there on the forest floor.
It is a war zone. It really is. But let's start with the star of the show. If I am walking through a mixed forest, say in North Carolina or up in Quebec or even down in Mexico, what am I looking for?
You're looking for a beacon.
Because this thing doesn't exactly blend in.
No, it wants to be seen. Amanita Jacksonii is a brilliant fire engine red, often fading to orange or yellow at the edges. But before you even see that red cap, you have to understand how it enters the world.
The emergence.
It's incredibly dramatic. You're talking about the egg stage.
Right, the egg stage.
And this is the defining feature of the Amanita genus. And it's where that alien vibe really comes in. The entire mushroom starts its life underground, completely encased.
Encased in what?
In a white, leathery membrane called a universal veil.
Universal meaning it covers everything.
Everything. The cap, the stem, the gills, it's all packed inside this white oval. To a forager, or just a casual observer, it looks exactly like a reptile egg or a puffball mushroom poking out of the soil.
And then it hatches.
It literally bursts. As the mushroom inside absorbs water and expands, it just tears through the top of that white veil.
That's an amazing image.
It is. The red cap pushes out like an alien emerging from a pod. And this is the crucial part, the mechanics of that break.
Because the veil doesn't just disappear into thin air, right? It's tissue.
Exactly. The veil is physical tissue. When the mushroom breaks out, the bottom part of that eggshell stays in the ground.
Okay.
It forms a cup around the base of the stem. We call this the volva.
Volva.
Volva. Correct. And if you take nothing else away from this conversation regarding safety or identification, let it be the vulva.
This is the number one thing.
It is the number one thing. It looks like a loose white sack that the mushroom stem is sitting in, like it's wearing a shoe or standing in a little bag.
One of the field guides we looked at described Amanita Jacksonii as a textbook mushroom. And I thought that was interesting. It has all the parts a diagram would show, which is actually kind of rare.
It's so rare. Usually mushrooms are missing something, you know.
Yeah.
The ring disintegrates or the base is indistinct.
But not this one.
This one is the perfect specimen for Anatomy 101. You have the cap on top, you have the gills underneath, you have a ring on the stem, we'll get to that, and you have the cup at the bottom.
The complete package.
Getting all four on one specimen is the complete package. It really is.
Okay, so let's walk up the mushroom then. We've established the white cup at the base, the vulva. What's next? What does the stem itself look like?
So the stem, or the stipe, is slender and yellow. But it's the texture that really gives it away. It's not smooth.
Okay, what kind of texture?
It often has these distinct orange fibers or chevrons.
Chevrons, like little V shapes pointing up.
Yeah, exactly. Or zigzags. Think of them as stretch marks.
Oh, that's a good way to put it.
It is. Because the stem grows so rapidly, it elongates so fast to lift the cap up for spore dispersal that the outer skin, the cuticle, it just stretches and breaks into these little snake skin patterns.
So it's a sign of its rapid growth.
It is, and it's very distinctive. If you see a smooth white stem on an Amanita, you should be worried. You want a yellow stem with these orange fibrous patterns.
And then there's the ring. You mentioned a ring.
The annulus, this is the remnant of the second veil.
Oh, wait, hold on. There are two veils. I thought there was just the one universal one.
Nope. Nature loves redundancy, apparently. The universal veil covers the whole egg from top to bottom.
Right.
But there's also a partial veil that just covers the gills. It connects the stem directly to the edge of the cap. Its job is to protect the delicate gills while the spores are maturing.
So it's like a protective sheet just for the important parts?
That's it. So when the cap opens up like an umbrella, that partial veil has a rip too.
And what's left of it is the ring.
It rips away from the edge of the cap and falls back against the stem, forming a little skirt. On Amanita Jacksonii, this skirt is usually yellow or orange, and it's very delicate. It just hangs down like a collapsed tent.
Okay, so we have the white cup, that's the universal veil at the bottom, and the yellow skirt, the partial veil on the stem. Nature really wraps this thing up tight before it's ready.
It protects its investment. A lot of energy goes into making spores.
So now the main event, the cap. We said it's red. Is it always red?
Ideally, yes. A fresh young specimen starts a deep crimson red. But pigments in nature are unstable.
Sun and rain take their toll.
Exactly. Sun, rain, and just time break them down. So typically the center of the cap stays red, but as you move toward the edge, it fades to orange and then maybe to yellow at the very margin.
So it creates a gradient.
A beautiful gradient.
Like a sunset.
It's a very apt description.
But there's a structural feature on the cap that is even more important than the color, the striations.
Striations. Those are the grooves, right? The little lines. Yes. If you look at the margin, the very edge of the cap, you will see distinct radial lines. It looks like the edge of a pie crust or the lines on a scallop shell.
You can feel them with your finger.
You can. These are called striations. The cap is physically thinner there, and you can almost see the lines of the gills right through the flesh of the cap.
And why is this a safety feature? Why is this so important?
Because the deadly species, the destroying angels and the death caps, they generally do not have these pronounced striations. Their caps are usually smooth and solid right to the edge. If you see a smooth margin on a white or greenish Amanita, your alarm bell should be ringing. Jacksonii has grooves.
Okay, I want to recap the checklist because this is dense and honestly terrifying if you get it wrong.
It is. Let's do it.
Number 1, vulva. A white, sac-like cup at the base. You have to see it.
You must see it.
2, stem.
Yep.
Yellow with orange snakeskin patterns or chevrons.
Correct.
3, ring. A delicate yellow skirt on the stem.
Yep.
And 4, cap. Red or orange with distinct grooves on the edge. The striations.
Perfect. And number 5, the gills.
Right, the gills. We forgot the gills.
We can't forget the gills. The gills of Amanita Jacksonii are yellow.
Not white.
Not white. They can be a pale yellow, a buttery yellow, sometimes a distinct lemon yellow, but they're yellow.
And the bad guys?
Pure white. The destroying angel has white gills. The death cap has white gills. If you flip a mushroom over and the gills are snowy white, you stop. You do not pass go. You just admire it and walk away.
It seems like yellow is safe, white is danger is a decent rule of thumb, but only within this specific context.
Oh, absolutely. Within the context of North American Amanitas, when you're comparing the Caesar group to the destroying angels, yes.
Okay.
But please, listeners, do not apply that rule to all mushrooms in the forest.
Right, that's a disclaimer we need to put in huge flashing lights.
Yes. There are plenty of deadly yellow mushrooms in other genera, but for this specific high-stakes comparison, the yellow gills are a key differentiator.
Okay, last identification point. Where are these growing? If I see this red beauty growing out of a rotting pine log, do I pick it?
You absolutely do not. That is not Amanita Jacksonii.
Because of its diet. We're going to get into the genomics of this later, but this is a key field mark.
It is. Amanita Jacksonii is a mycorrhizal.
Which means?
It means it forms a partnership with living trees. It grows in the soil, usually under oaks or pines. Its mycelium is physically connected to the tree roots. It does not eat dead wood.
So it's a partner, not a decomposer.
Exactly. So if you see a look-alike on a log, it's a saprobe. a decomposer, and it is not our emperor. It has to be coming from the ground near its tree partner.
So we've established what it looks like, but why is it called the American Caesar? Is there a Roman Caesar?
There is Amanita caesaria. This is the original, the OG Caesar. It grows in southern Europe and North Africa.
And the Romans actually ate this.
Oh, they loved it. It was a favorite dish of the Roman emperors. I read somewhere that they called it boletus in ancient Rome, which is just so confusing for us now.
Right, because now boletus means a totally different kind of mushroom with pores instead of gills.
Taxonomy is a fluid language over 2,000 years. But yes, the Romans loved this mushroom. It was considered the cibus diorum, the food of the gods.
The food of the gods, wow.
The American version, Amanita Jacksonii, was long thought to be the exact same species. It looks almost identical.
But it's not.
It's not. Genetic analysis and some very subtle physical differences proved it's a distinct species native to North America.
So Amanita Jacksonii is our local royalty.
It is. But here's where it gets complicated, because of course it does.
Nature is never simple.
Never. It's part of what mycologists call the Jacksonii complex. It turns out there isn't just one. There's a whole cluster of similar related species in North America.
Right, I saw them mention the yellow Caesars.
Exactly.
Wow.
Not all of them are that fire engine red. If you are foraging in the southeast, especially somewhere like Arkansas or Missouri, you might see Amanita arkansana, the Arkansas Caesar.
And how is that different?
It's lighter. It's more of a smooth yellow-orange, doesn't get that deep crimson center that Jacksonii has.
But it still has the vulva and the striations and the yellow gills.
Yes. The critical structural features remain. It's the color palette that shifts.
And there was another one mentioned, Amanita Benningiana.
Yes. Named after Mary Banning.
Who was she?
She was an incredible self-taught mycologist in Maryland in the late 1800s. And when I say amateur, that's doing a lot of heavy lifting. She was brilliant, but as a woman in that era, she didn't have institutional access to universities or publishing.
So she was an outsider scientist.
Totally. And she described this slender Caesar, which is smaller and often has a yellowish-brown spot in the center of the cap. It's a more subtle species.
It's cool that they're finally naming these things after the people who actually studied them.
It is. Justice for Mary Banning. But for the forager, the point is this. Nature loves variety. While Jacksonii is the classic red one, you might encounter these cousins. They are generally considered edible and safe as well.
Provided you identify them correctly as part of this group, this section of the genus.
Provided you do your homework, yes.
But that homework is so important because we have to talk about the neighbors you don't want to invite to dinner.
The dark side of the family.
Yeah, the destroying angels.
Amanita bisporigera in the eastern U.S. These are, they're beautiful, they're elegant, pure white mushrooms, and they are absolutely deadly.
And they grow in the same woods at the same time.
Often side by side. I've seen them just feet apart. And to a novice eye, a young destroying angel emerging from its egg might look confusingly similar to a young Caesar before the colors fully develop.
But the angel is all white.
Correct. White cap, white stem, white gills. The whole thing is ghostly white. Jacksonii has the colorful cap and the yellow gills.
But you mentioned that color can be unreliable.
It can. Sometimes environmental conditions can bleach colors, or a mushroom can be deformed or old. This is why we say you need to check all the features. You can't rely on just one.
So if you see a white cup at the base, but then you see white gills and a pale cap.
You walk away. Immediately. No second guessing. And then there's the crazy cousin, the one everyone knows from emojis and video games and fairy tales.
Amanita muscaria. The fly agaric.
The red mushroom with the white warts.
It's iconic. It's probably the most famous mushroom image in the world, but it's a totally different beast from Jacksonii.
It is. Visually, what's the biggest giveaway?
The warts. 100% the warts. Remember the universal veil, the white eggshell?
Yes.
In Jacksonii, it breaks cleanly at the top and leaves that neat cup at the bottom. In muscaria, the veil is more fragile. It sticks to the cap as it expands and breaks into little white patches or warts.
So Jacksonii has a clean cap and a cup at the bottom.
And muscaria has a warty cap and, what does it have at the bottom? No cup.
It doesn't have a clean, sac-like cup, no. It usually has shaggy, concentric rings of tissue at the base of the stem. It looks messy at the bottom. So messy base, warty top.
That's a great shorthand for muscaria. Whereas Jacksonii looks neat, it's sitting in its clean little bag.
And chemically, this is the most important part. Oh, night and day. Amanita Jacksonii is a culinary mushroom. It has no known toxins. It's just food.
And Amanita muscaria.
It contains ibotenic acid and mucimol. These are psychoactive and neurotoxic compounds.
Right. One of our sources went deep into this. It mentioned that muscaria is used for its psychoactive effects, but it's not at all like the magic mushrooms people usually think of, the psilocybin ones.
No, it's a completely different chemical pathway. It's a different experience entirely. It affects the GABA receptors in the brain.
And what does that do?
It causes a weird mix of sedation and delirium, sometimes nausea, muscle twitching.
Hmm.
It's not generally described as a fun or recreational high in the same way.
It's more disorienting.
Very. It famously involves macropsia and micropsia Alice in Wonderland syndrome. Objects in the room can look huge or tiny. Perception gets very warped.
And there's that whole Santa Claus connection. Is that real?
Oh, the Siberian shamanism theory. Yes. It's a compelling theory, at
Yeah.
least. The idea is that in Siberia, shamans would consume Amanita muscaria for winter solstice rituals. And the local reindeer also love eating these mushrooms.
Wait, reindeer get high.
Apparently so.
Eh.
They actively seek them out. And here's the weird part. The active compounds are excreted in urine largely unchanged.
Uh-oh. I know where this is going.
So, well, there are historical and anthropological accounts of the reindeer eating the mushroom and then people drinking the reindeer urine.
No!
Yes. To get the psychoactive effects without some of the nausea-inducing compounds that the reindeer's liver filters out.
Nature is beautiful, but sometimes it is just truly gross.
It is. But think about the imagery. You have red and white mushrooms under pine trees. You have flying reindeer, because they're prancing around high. You have shamans dressed in red and white who enter the yurt through the smoke hole because the door is snowed in.
The chimney.
Exactly. Some anthropologists trace the entire modern Santa Claus mythos back to these ancient Amanita muscaria rituals.
That is wild. Absolutely wild. But okay, back to our Slender Caesar. We've established it's not the druggy one, and it's not the deadly one, it's the delicious one. But why? Why is it safe when its cousins are so dangerous?
This is where we get to open up the genome and look under the hood.
Right, the 2014 study.
In 2014, there was a study published in IMA Genome F that actually sequenced Amanita Jacksonii. It was part of a larger project to sequence a bunch of interesting fungi.
Why did they pick this specific mushroom to sequence? What was the goal?
They wanted to understand the evolution of the Amanita genus. It's such a diverse and puzzling group, right? You have deadly toxins in some, edibility in others. You have saprogues, which eat dead stuff, and mycorrhizal ones, which partner with trees. Jacksonii is a perfect pivot point to study all these different traits.
What did they find in its DNA?
They found a genome of about 30 million base pairs, which is fairly compact, encoding roughly 8,500 genes. But the really cool part was looking at the metabolic pathways, specifically for the colors.
The red and yellow pigments?
Exactly. These pigments are called betalains.
Betalains. Wait, is that what makes beets red?
Exactly. It's the same class of pigment found in beets and Swiss chard and some flowers. And it's actually quite rare in fungi. Most mushrooms use melanins or other pigments. But the Amanita genus, specifically the section that includes muscaria and our Jacksonii, figured out how to make betalains.
So Jacksonii and the toxic muscaria both use the same paint from the same paint store.
Yes. They share the genetic machinery to produce these pigments, which also happen to be antioxidants. But here's the kicker. The genome shows that muscaria also has the genes to produce the toxins, the ibotenic acid.
And Jacksonii doesn't.
The genes just aren't there. It has the color genes, but it lacks the toxin genes.
So it's like Jacksonii got the flashy paint job, but didn't install the weapon system.
That's a perfect analogy, and it raises a fascinating evolutionary question. Why keep the color? In muscaria, the bright red is probably a warning signal of aposematism. Don't eat me, I'm dangerous and will make you see weird things.
A clear warning.
But Jacksonii is delicious. So is it mimicry? Is it bluffing? Is it pretending to be dangerous? Or does the color serve another purpose entirely, like attracting specific insects for spore dispersal?
Or maybe it just never lost the paint job because it wasn't hurting it, evolutionarily speaking.
That's also a real possibility.
Yeah.
Evolution doesn't always optimize for everything. If the color isn't energetically expensive to produce, and it's not getting it eaten into oblivion, the genes might just hang around.
The genome study also talked about lifestyle. You mentioned it's mycorrhizal.
Yes. This is a huge evolutionary shift in the fungal world.
Huh?
Ancestrally, most fungi were saprophytes. They were decomposers.
The recyclers of the forest.
Right. They had this huge toolbox of powerful enzymes to break down cellulose and lignin, the tough stuff in wood.
And the study looked at that.
It did. The study compared Jacksonii's genome to a saprophytic cousin called Amanita thyracea. And thyracea still has all the genetic tools to digest dead grass and wood. But Amanita Jacksonii has lost those genes.
It's lost them, like deleted them.
The genome shows they're just gone. They literally deleted the ability to make certain kinds of cellulose. They forgot how to eat dead wood.
That seems like a really risky evolutionary move. I'm going to throw away my ability to digest the single most abundant food source in the entire forest.
It is incredibly risky unless you have a guaranteed food source, a sugar daddy, if you will.
The tree.
The tree. By becoming mycorrhizal, Jacksonii plugged itself directly into the tree's root system. The tree does all the hard work of photosynthesis, makes sugar, and then pipes that sugar directly to the fungus.
And what does the fungus give back?
In exchange, the fungus acts like a massive, super efficient extended root system. Its fine web of mycelium can gather water and minerals like phosphorus and nitrogen far better than the tree's roots can on their own.
So it traded its independence for a partnership.
Exactly. And once it made that trade, it didn't need the digest wood tools anymore, so evolution, being efficient, tossed them out. That's why you will never find a true Caesar mushroom growing on a dead log. It physically cannot eat it. It needs a living host.
That is so cool. It just fundamentally changes how you look at the forest floor. It's not just a bunch of individual things growing on the dirt. It's a network.
A deeply interconnected network,
Yep.
and that network is crowded. As we mentioned, the genomic study didn't just look at Jacksonii.
Right, I looked at the neighborhood, and it's a rough neighborhood.
It's a very rough neighborhood.
Yeah, I saw some of the other names in that study. Fusarium cercinatum. That sounds like a spell from Harry Potter, but I'm guessing it's not a good guy.
It is an absolute villain.
Yeah.
Fusarium cercinatum causes pitch canker disease in pine trees.
Pitch camp.
Pitch is resin. When this fungus attacks a pine tree, the tree tries to defend itself by bleeding resin copious amounts of it. It's a desperate defense.
And it doesn't work.
It doesn't. The tree basically bleeds to death trying to flush out the invader. The cankers just weep resin until the tree is exhausted.
Yikes.
And genetically, this fungus is fascinatingly weird because it has what are called dispensable chromosomes.
Dispensable, like it can just lose them and be fine.
Yes. In the lab, researchers found some strains just dropped an entire chromosome, I think it's chromosome 12, and just kept on living no problem.
How is that possible?
It implies that parts of its genome are like downloadable content or expansion packs. It can pick up or drop certain genetic toolkits depending on what kind of host it needs to infect. It makes it incredibly adaptable and dangerous.
That is terrifyingly adaptable. Wow. Okay, who else lives in this forest?
Then there's Leptographium procerarum. This one is interesting because it partners with a beetle.
The red turpentine beetle, right?
That's the one. This is a classic invasion team. The beetle bores into the tree and it carries the fungal spores with it, inoculating the tree as it goes.
So they're a team.
A destructive team. The beetle gives the fungus a ride, and the fungus helps break down the wood, making it easier for the beetle larvae to eat. But here's the really diabolical twist.
There's more.
The research suggests that the fungus might actually manipulate the tree's chemistry to produce more turpentine smells.
But why would it do that?
To attract more beetles, it creates a destructive feedback loop. Come here, this tree is weakened and open for business. It turns the tree into a beacon for its own destruction.
That's pure evil genius.
It is. And then you have Ceratocystis albifundus. This one causes a deadly wilt in acacia trees in Africa, and its strategy is scent. It produces this sweet, fruity odor
To attract us.
to attract insects. It smells like rotting fruit or fermentation. The insects, like fruit flies and beetles, think they're getting a meal.
Instead.
They land on the fungus, get covered in its sticky spores, and then fly off to the next healthy tree, acting as unwitting delivery drones for the pathogen.
It's all just chemical warfare out there, isn't it?
It really is. So when you see that beautiful, innocent-looking red Amanita Jacksonii, just remember it's sitting right in the middle of this invisible battlefield. Pathogens are attacking the trees, beetles are drilling holes, other fungi are trying to poison each other, saprobes are quietly dissolving the dead leaves,
And the Amanita is there, hooked into the roots.
helping the tree survive so it can keep getting its sugar fix.
It really is the good guy in this story.
In this context, essentially, yes, it's a mutualist, a protector in a way.
Okay, let's pivot from the war zone to the dinner plate. Let's talk about the part everyone is waiting for, eating it.
Ah, yes, mycophagy, the culinary appreciation.
The sources are pretty unanimous that this is a top-tier edible. One source quoted someone saying it tastes like fancy cheese.
That is a very specific and intriguing description. I've also seen refined, nutty, cheesy. It has a complex flavor profile that isn't just mushroomy. you know?
And what's really unusual is that, unlike almost every other wild mushroom we talk about, people eat this one raw.
Yes. This is a huge exception to the rule. In Italy, Amanita Jacksonii is famously served as a Carpaccio.
So just sliced thin.
Just sliced paper thin while it's fresh, arranged on a plate, and drizzled with a really good olive oil, a little squeeze of lemon juice, maybe some sea salt, and shaved Parmesan. That's it.
That sounds incredible. But also, my brain is screaming, don't eat a raw wild mushroom. We're always told to cook them thoroughly to break down chitin and any potential toxins.
And 99% of the time, that is the correct rule. But the Caesar group is a famous exception. It's tender enough and, more importantly, safe enough to eat raw. Though lightly sautéing it in butter is also, heavenly.
I bet. Now, one of our sources mentioned a preference for harvesting them in the egg stage or just as they're opening. Why is that?
2 reasons: texture and bug control.
Ah, the bugs.
As the mushroom fully opens and creates that big flat cap, the gill can get a bit soft. But more importantly, insects love these mushrooms too.
Oh, right, if it's delicious to us.
It's a 5-star hotel for fungus gnats and their larvae. By harvesting the egg or the young button, you're getting the mushroom when the flesh is perfectly firm and pristine before the bugs had a chance to move in and lay their own eggs.
But there's an ethical or maybe more of a safety dilemma with the eggs, isn't there?
The huge safety dilemma. This is probably the most contentious issue in foraging this species.
Let's break it down.
Okay. When an Amanita is in that egg stage, it is completely encased in the white universal veil. If you find one, and you're an expert, you can slice it open vertically.
And you can see inside.
You can see the baby mushroom inside the embryo, so to speak. Inside a Jacksonii egg, you will see a yellow or orange outline of the cap. The pigment's already there.
Okay, that makes sense.
Inside a Bisporgera egg, a destroying angel, you will see pure, ghostly white.
So you can tell them apart?
You can if you cut every single one open. But from the outside, a white egg in the ground is a white egg in the ground. It could be a Caesar, it could be a death angel, it could even be a young, harmless puffball mushroom. Just don't know.
So picking eggs is playing Russian roulette for anyone who isn't a top-level expert.
It is extremely high risk for beginners and intermediates. If you mistake a death angel egg for a Caesar egg and eat it, that is a fatal mistake. There's no coming back from that poison.
So what's the advice?
The advice from most experts is, let it open. Let it reveal its colors and its features. Don't be greedy. Wait until you see the red cap, the yellow gills, and the striations. Be patient.
Patience is a survival skill in mycology.
It's the #1 survival skill.
There's a historical story about this exact kind of confusion, right? The poisoning of Emperor Claudius.
Yes, the classic Roman whodunit. It's a fantastic and grim story.
So the setup is Agrippina, the wife of Emperor Claudius, wanted her son Nero to take the throne. But Claudius was in the way.
He was. And Claudius loved his Caesar mushrooms, Amanita Jacksonii.
It was his favorite food.
He couldn't get enough of them. So the story goes that Agrippina conspired with the royal taster, or maybe the cook, the accounts differ, to slip some death caps, Amanita phalloides, into a dish of sautéed Caesars.
Which is deviously clever.
It's the perfect crime. The death cap is also an Amanita, so it has a similar texture, and when they're chopped up and cooked, they'll be very hard to tell them apart. And the death cap toxin, amanitin, is completely heat stable. Cooking doesn't destroy it one bit.
So poor Claudius is eating his favorite fancy cheese-tasting mushrooms.
And he enjoys his meal. He probably complimented the chef. And that's the truly cruel part of Amanita poisoning. There's a delay, a latency period.
Right, you don't get sick right away.
No, you feel perfectly fine for 6, 12, sometimes 24 hours. Then, violent gastrointestinal illness, vomiting, cramps. It's awful. But then, you might even feel better for a day.
The honeymoon phase.
The false recovery. But all the while, the amatoxins are silently, systematically destroying your liver cells.
And he died, and Nero became emperor, and the rest is, well, very messy history.
All because of mushroom mix-up. Intentional, in this case.
Whoa.
But it serves as a powerful warning. Even emperors get tricked by amanitas.
Wow. So for our listener who is maybe hiking in the woods this weekend, they see a beautiful red mushroom. They think, maybe, just maybe, this is the emperor. Let's do a rapid-fire checklist one more time.
Okay, let's drill it in. 1, color. Is the cap bright red or orange? Maybe fading to yellow at the edge. 2, striations. Does the edge of the cap have distinct grooved lines, like a pie crust?
Check.
Gills. Look underneath. Are they yellow? Not white, not cream. Yellow.
Crucial check.
4, stem. Is the stem yellow, and does it have those orange fibrous snakeskin patterns.
Sure.
5, vulva. And this is the big one. Is there a distinct white sac-like cup at the very base? And you might have to gently dig a little to see this.
Dig, don't cut.
Always dig, don't cut. And 6, habitat. Is on the ground, under oaks or pines, not growing on a log.
Okay. If you can confidently check all 6 of those boxes.
You very likely have Amanita Jacksonii or one of its safe close siblings in the Jacksonii complex.
But if you are even 1% unsure about any of them,
Take a picture, admire its beauty, and leave it alone. As one of our sources so perfectly said, if you can't define striations or vulva, you shouldn't eat it.
that is the best advice. Now, before we wrap up, I want to loop back to something we touched on earlier. The complexity of the other Caesars in North America. We have the red Jacksonii, but there's a whole group of them.
Yes, this is for the advanced class. This is when you've mastered the basics and you want to appreciate the nuance.
Tell me again about the Arkansas Caesar, the Arkansana.
So if you are in the southeast, especially Arkansas or Missouri, you might see Amanita arkansana. It doesn't get that deep crimson red of Jacksonii. It starts more orange-yellow, and it stays that way. The whole cap is more of a uniform soft orange.
But all the other safety features are the same.
Exactly. Yellow gills, striated margin, vulva at the base. All the non-negotiables are there. It's just a different colorway.
And Mary Benning's slender Caesar, Amanita benningiana.
That one is fascinating because it often has this brownish, almost bruised-looking spot right in the center of its yellowish cap. And as the name implies, it's generally smaller, more slender and delicate than Jacksonii.
It's like spotting the difference between a downy woodpecker and a hairy woodpecker. You really have to start looking at the fine details.
That is the perfect analogy. And this is where citizen science is becoming so important.
How so?
Well, one of our sources was a red list entry, an assessment trying to track the population of Jacksonii and its relatives. They rely heavily on observations from regular people using apps like iNaturalist.
So my photo could actually help.
Absolutely. Because this Jacksonii complex is still being unraveled by geneticists. Photos and location data and notes on which trees they were near, all from regular people, are actually helping scientists define where one species ends and another begins.
We're helping map the family tree.
You are. We need to map these yellow Caesars versus the red Caesars. Are they strictly separated by geography? Do they prefer different tree partners? We're still learning, and every observation helps.
I love that. The forest is still full of mysteries we can help solve.
It is. We think we have it all cataloged in our field guides, but we're just scratching the surface of the genetic reality.
Okay, as we get ready to leave the woods here, I want to talk about mushroom blindness.
Oh, that's an interesting term. I like that.
It's something one of our sources touched on, the idea that once you learn to spot Amanita Jacksonii, you start seeing it everywhere. But before you knew what it was, you probably walked past 100 of them and your brain just registered red blob and moved on.
That is the reticular activating system in your brain at work.
Yeah.
It's a real neurological phenomenon. Once you give your brain a search image, once you know the pattern of red cap plus yellow gills plus white cup, your brain highlights it for you. It literally pops out of the background noise of the forest.
It's exactly like when you learn a new word and then you start hearing it 3 times a day.
Precisely. And Amanita Jacksonii is a great entry point for curing mushroom blindness because it is so distinct. It's not a cryptic little brown mushroom hiding in the leaves. It screams, "Look at me,"
It's a gateway mushroom.
in the best possible way. It forces you to learn the right habits. It forces you to look at the vase for the vulva, to look at the gills for the color, to look at the cap edge for the striations. It trains your eye to look for the details that matter for all mushroom identification.
And in doing so, it connects you to this deep history we've been talking about. The Romans, the genomic evolution, the complex, invisible web of the forest floor.
It really does tie everything together. It's a survivor, a partner, a beauty, and a delicious meal, but only if you earn it with knowledge and caution.
So what does this all mean for us? What's the big takeaway?
I think it's a powerful reminder that nature's nuanced. We like to categorize things into simple boxes, good or bad. Amanita is the bad genus.
Right, the scary one.
But within that, you have this absolute gem. You can't judge a mushroom by its family name alone. You have to look at the individual. You have to understand the specific traits that make it what it is.
And you have to respect the chemistry.
Always respect the chemistry. The difference between a delightful dinner and a liver transplant is, in this case, a few genes and a few distinct molecules. The margins are incredibly fine.
And maybe that's the final thought to leave our listeners with as they walk out of the woods. Why did this mushroom keep its bright colors? You mentioned mimicry earlier, and it's a fascinating idea.
It's a provocative thought, isn't it? Is Amanita Jacksonii wearing the uniform of its toxic cousin Amanita muscaria to stay safe? Is it shouting, "I'm red, I'm dangerous, leave me alone," even though it's a complete bluff?
A sheep's in wolf's clothing.
Exactly. Or is it something else entirely? In the tropics, many unrelated mushrooms have evolved to be bright red to attract certain kinds of spore-dispersing beetles. Maybe Jacksonii is an evolutionary holdover from a tropical ancestor, and its color is a message for an insect that isn't around anymore.
Or maybe, just maybe, it's just showing off.
In nature, beauty usually has a budget. If you're spending precious energy on making pigment, it's almost always for a reason. We just might not know the reason yet, but it's there, hiding in plain sight.
Something to ponder on your next hike. Next time you see a flash of red in the ferns, stop. Crouch down, check the base, check the gills, look for the striations, and you might just meet the emperor.
Just remember what you said, dig, don't cut. You have to see the vulva.
Always dig. Thanks for joining us on this deep dive into the fungal web. We'll see you in the next one.
Stay curious.